Hi everyone. Welcome to my YouTube channel. Today we are going to start electron microscopy under Unit I Microbiology. In the previous video we have studied about dark field and phase contrast microscopy. What is electron microscopy? In this we make use of stream of electrons duly deflected either by an electrostatic or by electromagnetic field for magnification of objects. Resolution is higher. Internal structures of cells and viruses can be examined as electron possess 100,000 times smaller wavelength than visible light. Electron microscopy can be further divided into two types: transmission electron microscopy and scanning electron microscopy. Transmission electron microscopy uses extremely fine focused beam of electrons from electron gun that penetrates via a specially prepared ultrathin section of specimen. Column containing lenses and specimen must be under high vacuum to obtain clear image. Resolution limit 0.2 micrometer. Application of TEM is to see information about interior of cell and arrangement of proteins, lipid layer. Scanning electron microscopy is used to examine surface of various organism. It shows primary electron sweeping across specimen together with knock electron emerging from its surface. Resolution 7 nanometer or less. Specimen preparation: microorganism must be fixed, dehydrated, dried to preserve surface structure and prevent collapse of cells on exposure to SEM's high vacuum. See the diagram on the screen to know about the information that we can get by using transmission and scanning electron microscopy. Electron microscopy advantages are magnification 10,000 times better. Diverse applications in research, agriculture. Disadvantages: costlier instrument to build. High maintenance required. Difficult to relocate. Skilled personnel can do it. Thanks for watching the video. Please like, share and subscribe my channel for latest updates. If you have any query then you can write it in comment box.